Hello, everyone. Hello. My name is Assia, and I'm a year one student. Today, me and my friends are going to present biochemistry quiz one revision. So we'll start by chapter 3. All right. So to start, why is water essential for life? What can you guys tell me about this? Any ideas? Yes. Okay. Anything else? Yes. Yes, that's what he said. Okay. Any other ideas? No. All right, so water is essential for life thanks to several of its properties. So we have properties such as chemical stability, its remarkable solvent properties, like you guys said, its role as a biochemical reactant, and its importance in hydration. Water also has an important cohesion within the same molecules. So this also contributes to how it's a good solvent. Now for the structure of water. So you guys might know this already. Water has a V shape, so it's the molecule H2O is bent. You guys know why is it bent? What's the reason behind this? Yes? Yes, that's right. Oxygen has two pairs of unshared electrons, and because of their repulsion, the electron pair geometry is tetrahedral, but the molecular geometry is bent, as you can see in this diagram. So something else that is very important about the structure of water is its polarity. So what is polarity? What can you tell me about it? Yes? Okay, anything else? Yes? Yes. Okay, so as oxygen is more electronegative than hydrogen, oxygen has what we call a partial negative charge, while hydrogen has a partial positive charge. So that means that some of the electron that is shared between oxygen and hydrogen is going to be closer to oxygen than hydrogen. And because, as we mentioned earlier, the molecule is bent, the partial positive charge will be on one side and the partial negative charge will be on another side. So it's going to be what we call a dipole. So this is polarity. It's when in the same molecule you have one side that is partially positive and one side that's partially negative. But the entire thing is neutral because it's a molecule. So, all right. So a little question for you guys. Which of the following properties of water does not contribute to the fitness of the aqueous environment for living organisms? Any ideas? Yes? Any other suggestions? Yes? Okay. Someone said B? All right. So the correct answer is E. So the density of water is greater than the density of ice. This is why ice floats on water. And water does have a high heat of vaporization compared to other similar compounds. Water's boiling point is much higher. But, however, water does not have a very low molecular weight. All right. Next, we'll move on to non-covalent bonds. So, do you guys know the difference between non-covalent bonds and covalent bonds? Okay. Can anyone explain? Yes. So, covalent bonds are intramolecular. So, within one single molecule, the interactions between the different atoms are called covalent bonds. Non-covalent bonds can be within the same molecule, but they're usually between several molecules, so intermolecular. So we have different types of non-covalent bonds. Among them, we have ionic interactions. They're also called electrostatic bonds or salt bridges when they're found in proteins. So ionic interactions is, as we see here, for the NaCl, the electron of Na is taken by the Cl, which has a negative charge, and the Na has a positive charge. Now to another type of non-covalent bonds, which is hydrogen bonds. So hydrogen bonds is one of the main reasons for the high cohesion of water molecules. And that is because water molecules stick together thanks to the partial positive and negative charges. So as you can see here, the O that has a partial negative charge is going to form a hydrogen bond with the H that has a partial positive charge. And this can happen with other atoms other than O. So do you guys know which other atoms can cause? Yes. Yes. If you have a molecule with an NH or FH bond, you can also observe hydrogen bonds there. All right. And for the last type of non-covalent bonds, van der Waals forces, also called hydrophobic interactions. Do you guys know why they're called hydrophobic interactions? Okay. And you find them especially in molecules, in lipid molecules that don't mix up with water. And we have three types of van der Waals interactions: dipole-dipole, dipole-induced dipole, and induced dipole-induced dipole. So, do you know which ones are caused by what from the three boxes underneath? Yes. Mm-hmm. Yes. All right, that's correct. So, as the name indicates, dipole-dipole is when both molecules are dipoles. So there's a permanent partial positive charge and a permanent partial negative charge. Dipole-induced dipole is when only one of them has entirely permanent partial charges, and induced dipole is instantaneous. All right. Now to test whether you've been listening. Which of the following describes the most likely way of which two water molecules would interact? Yes? Okay. Any other answers? Yes? Okay. Everyone agrees on B? All right. The answer is B. So a hydrogen bond will form between the hydrogen atom of one water molecule and the oxygen atom of the other water molecule. Next. So where would the hydrogen bond form in this molecule? All right. Any other answers? B? And the answer is B. Correct. All right. Now, which intermolecular forces are present in molecules of this type? Which answer? Dipole, dipole. Anything else? Who said C? Okay. And yes, it is C. Yes. Any questions about this? Okay. So the London dispersion forces are found in any molecules because they're instantaneous. They come and go. They're always present. For the dipole forces, it's because we can see that there are wait, hold on. Now I'm not sure of the answer anymore. Can yes. Mm. Yeah. Yes, that is true. So it's not a hydrogen bond because we don't have any FH or OH or NH bonds. So yes. Now, can you guys tell me the names of these three non-covalent bonds, starting with this one? Yes, hydrophobic interaction. What about the green one? Hydrogen bonds. Right. All right. For the blue one. So this is a hydrogen bond because we can see OH and O. So the H that has a partial negative charge and the O that has the H that has a partial positive charge and the O that has a partial negative charge. Here you have electrostatic interactions, so ionic interactions, because we can see here a negative charge and here a positive charge, and over there hydrophobic interactions. All right. Any questions on non-covalent bonds in general? No. All right. Now, on to part 3.5 by my colleague.
Hello guys, so I'll be covering 3.5, the ionization of water. And then we'll go back to 3.4, don't worry. Okay, so before we start, I have a different way of kind of doing things. So we'll test this in rounds. Each round has a different concept within the topic of 3.5. We'll go through the questions using the process of elimination. So we'll go through A, B, C, D till the end. And I want us to go through it together through the thinking process that I hope that you can use while you're going through your exam questions. And then lastly, for each round, I want a volunteer to come help me on the stage. Is that okay? Okay, let's start. Round one, can I have a volunteer? Who can volunteer? Yalla, come. Okay, بسرعة. So here is the round one. The concept that we're testing is the ionization of water. What are the two ions formed when water ionizes? Okay, who can tell me? Yeah? B, hydrogen ion and hydroxide ion. Okay, so these are the two ions that are going to be formed. Now, can you tell me what's your name? Qasim, is this type of reaction, the ionization of water, is it reversible or irreversible? B. Perfect. So this over here is the concept of the ionization of water. We're producing hydrogen and the hydroxide, and as Qasim said, this is a reversible reaction. We're disassociating it. From its name, we're forming the ions that form water. Give a round of applause to Qasim. Okay. Moving on to the second part. I think it's easier.
Let's introduce the reward system.
Oh, صح, the reward. Thank you, Qasim. So anyone who comes and helps me in the round gets who wants to help me in the second round? Who wants to help in the second round? Come on. Perfect. So over here I have water, and I'm going to add a proton onto the water. So hydrogen ion is the same thing as saying I'm adding a proton. So if I add a proton to water, what will I produce? Can you tell me?
Hydronium.
Is he correct?
Yes.
Round of applause, please. But not too fast. A hydronium ion, is it? Does it have a structure of H3O plus? Okay, that's correct. Is a hydrated hydrogen ion? It's not?
Yes.
Because this is a hydrogen ion, right? Where's the hydrogen ion? I'm adding water onto it, so I'm hydrating the proton or the hydrogen ion. So therefore B is correct. Is C correct?
Yes.
Yes, because we just said if I say hydrogen ion, I also mean it's a proton. D is the usual form of one of the dissociation products of water in a solution. Who said no? It is correct. So therefore, what is the answer? All of the above. Thank you so much. Don't forget Yasser, don't forget to give him the... Perfect. Round 3, who can help me out with this one? Go ahead. And then you'll come for round 4. Okay, so round 3, the concept that we're testing, guys, sorry, settle down, please. The concept that we're testing for round 3 is the ion product of water, okay? That's the same as saying the equilibrium constant of water. What's her name?
Abdullah.
Abdullah. What is the ion product of water at 25 degrees? So if I say 25 degrees, what does that indicate? Room temperature. And also, I could add to that an atmospheric pressure at the normal atmospheric pressure. So which one of these is the correct answer? Is it A?
No.
Is it B?
No.
Is it C? No. So the answer is B. Bravo. But Abdullah, what is the ion constant? What is the ion constant of water?
Is it like a proton with hydrogen? Oh,
The concentration of the
hydrogen?
hydrogen times the concentration of the?
Oh,
Bravo. Well done. So guys, this is the ion product of water. It's essentially the concentration. I'm looking for the concentration of the hydrogen ion times the concentration of the hydroxide ion. That's how I'm going to get the Kw. And then at a normal room temperature and atmospheric pressure, it's going to be 1 times 10 to the power of negative 14. Who's going to be my volunteer? I think you wanted to be the volunteer for round 4? Yeah? Okay. Round 4 is going to be about acids and bases. According to Bronsted-Lowry definition, what is an acid? Let's do again the process of elimination together. Is it a proton donor? خلاص يبقى كده خلاص. It's a proton donor. صح. طب ده معناه انه what is the base? It's a proton acceptor. برافو عليكم guys. You guys are ready to go. So yes, an acid is a proton donor and then a base is a proton acceptor. Okay? And this over here is a diagram visualizing this. I'm an acid. I need to have a proton in order to donate it. Now, if I'm going to donate, right? What is going to be produced when I donate my proton? A conjugate base, exactly. So, and this reaction is always reversible. Hence, same thing with a base. This base over here, once it gains the proton, after the reaction, what is it going to give me? It's conjugate acid. Perfect. Perfect. And this concept will take us to pKa values. تعال. Yeah. معلش. What's your name? Yahya. Based on the table, which of the following acids is the strongest? Now,
What?
Yahya, which of these is the strongest acid based on this table?
Hydrochloric acid.
So, Yahya is saying hydrochloric acid.
لا hydrochloric acid.
Okay, hydrochloric, last answer. Is it hydrochloric acid? Yes. Why? Lowest pKa value. That's correct. So what is then the weakest acid? So it's going to be why? Highest pKa value. That means that when I have a low pKa value, I am a stronger acid. When I have a high pKa value, I'm a weaker acid. Give it up for Yahya. And this is again kind of telling us the same concept that we went over. pKa, as when it increases, the strength of the acid decreases. And this is the how we get the pKa. The pKa comes from the Ka, negative log of the Ka. And the Ka, again, we obtain that from the dissociation. And that same concept for Ka is the Kw, which we looked into. Perfect. Buffers. Can you come help me out on this? What's your name? Yeah. لا لا عادي. You got this. لا تجيبي صح. We're gonna help you. Don't worry. يلا. What's your name? Sarah. Give it up for Sarah, everyone. يلا Sarah. What is a buffer commonly composed of? Is it a strong acid and its conjugate base? Is this true? Is it a strong base and its conjugate acid? Is it a neutral compound? Is it a weak acid and its conjugate base? You got it right? So buffers, guys, commonly, it's a weak acid and its conjugate base. However, it could also be a weak base and its conjugate acid. Okay? So this is kind of an explanation. So I have a buffer over here. If I am going to add an acid to that buffer, which part of the buffer is going to react with the acid that I'm adding? The conjugate base. Now, if I'm going to add base onto my buffer, which part is going to react? The weak acid. Exactly that. So the buffer is going to work to neutralize whatever is being added to it in order to maintain that equilibrium. So this is the concept of buffers. And again, one trick to kind of identify, now they could not give you this is a buffer is a weak acid and its conjugate base. They could give you an example and tell you identify مثلا the weak acid مع الconjugate base. So how do you identify? Usually the difference between them is going to be a proton. Let's go back to the definition. An acid is a what? A proton donor. So realistically, I'm literally going to remove that proton and I'm going to get its conjugate base. Does that make sense? Perfect. Moving on to round number, and that's the same concept that we just went over. Round number 7, who wants to help me out? Islam? Yeah. In the titration of phosphoric acid with NaOH, why are there multiple buffering regions? Islam, is it A, phosphoric acid is a strong acid? Is phosphoric acid a strong acid?
No. Phosphoric acid starts at pH value of 2, which is pretty low, so I'd say it's a strong acid.
You say it's a strong acid? Guys, is it a strong acid? No. It's not a strong acid. Okay, is it the base neutralizes the acid all at once?
No.
No. Why? Because we have different levels of ionization, so the acid releases all protons at the same time? No. Therefore, the correct answer is going to be B. Phosphoric acid has multiple ionizable protons. Then what is the equation, or what is the component of phosphoric acid? H3PO4. So? H3PO4. For how many protons can it donate? 3. Therefore, I have multiple ionizable protons, and that's why we have a stepwise curve. Does that make sense? Yes. Give it up for Islam, everybody. Thank you. And the last concept that we want to test for 3.5 is going to be physiological buffers. Who wants to be the last volunteer? Huh? Come on. Okay, خلاص. We'll do it together. Which of the following represents the bicarbonate buffer reaction? Is it A? Yes. Why is it not B? Or C or D? Yeah, basically it's A, because this over here is the bicarbonate acid. Whether I'm going to add an acid to it or I'm going to add a base, this is going to be the buffer equation. So this buffer equation is one of the things that you need to know. This mainly controls essentially the acidity and the basic of the blood. So this is a very important physiological buffer. But what is the enzyme that catalyzes this reaction? Are you going to let him answer, or are you going to answer? What is the enzyme?
I'm not sure, but is it carbonic anhydrase?
Carbonic anhydrase. Well done. So, it is carbonic anhydrase, and again, this is the way that buffers work. Buffers would do anything to try to neutralize any change that occurs in the blood. Hence, if acidosis happens, what is acidosis? The acidity of the blood increases. So that means what's going to happen, the bicarbonate ions is going to react with the increased acidity. More hydrogen ions giving us this carbonic acid. This carbonic acid can then later dissociate into water and CO2, and vice versa. And that's it. Thank you. And of course, shout out to Claude. Yeah. I sent some Arsenal fans, Man United fans. I gotta leave. I'll stay for 50 minutes.
اشتغل؟ Hello? تمام. Okay. Okay.
Okay, hello everyone. How are you all doing so far? Is your brain intact? Okay, are you all ready? Tamam. So I'm Rand, I'm a Year 2 medical student, and I'm going to be talking about the solvent properties of water. Okay? So this is going to be an interactive session. I want you all to answer. And for the first thing we're going to be doing is, it's kind of like a quick revision. What we're going to do is basically we're going to relate the following terms to water. So I want you guys to give me an example, give me a story, whatever you want to say, say it. So for the first one, polarity. How does this relate to water? Whoever has an answer, like say it out loud. It has unequal. Where did you say? Exactly. Okay, it has an unequal distribution of electrons. So what does that mean? Exactly. Correct. Yeah. All of your answers are correct. Good job. So as you can see over here, water has, the electrons are distributed in a manner where it gives a partially positive charge to the hydrogen and a partially negative charge to the oxygen, okay? And now this is going to relate to hydrogen bonding, which is number 2. Who can give me, I don't know, a story, an example relating to water and hydrogen bonding? Okay, water molecules bond with hydrogen bonds. Anything else? Guys, you need to answer. Anything you have to say, Adi, say it. Correct. Yeah, exactly. It's important in the formation of proteins. Anything else? This side. You guys haven't said anything. Correct. Okay. Exactly. So good job, everyone. Basically, hydrogen bonding, as Assia said, is an attraction between the partially positive charge on the hydrogen and the partially negative charge on the oxygen. And it could also be a nitrogen molecule or a fluoride molecule. Is that correct? Tamam? Okay. Now we're moving on to number 3. How does water incorporate with proteins or with cellular components? Anyone have anything to say? Someone else. Okay. Do you have anything to say? Good. Okay, correct. Guys, settle down, please. Correct. Those are some of the functions of water in the cell. As we know, water composes cells in every single being. And what it does is that it forms like a protective layer or support around the proteins, okay? Guys? Okay? So what this does is that basically it's a protection. It can form as a communication medium between the proteins in the cell and other cellular components like the mitochondria. Something else it can do is maybe, like facilitate biochemical reactions, okay? So that's one of the functions of water inside the cell in relation to proteins, all right? Now, last but not least, how is water versatile? Why is it versatile? Yeah, that's correct. So it has many functions. What else? I can hear something here too.
It's all of the body, most of the body.
Metabolic reactions. Correct. That too. You were saying something? I can't hear you. Correct. That is correct. I was going to ask about that next. But he mentioned that it has a high specific heat capacity, so it can hold in a lot of heat before it starts to give it out. And as you know, water is a solvent that basically nearly all of the solutes in the world can dissolve in it. Not all of them, but most of them. And that's why it's called the solvent of life, okay? So now we're going to move on to questions. So I'm going to give you guys a few seconds. We're going to read the question, we're going to answer, and we're going to see what percentage of you get the answer. And we're going to discuss the options that are not the answer, okay? So the first question says, a true statement about hydrophobic interactions is that they, A, are a driving force in the formation of micelles of amphipathic compounds in water. Do you guys think this could be the answer? No? Okay, we'll move on. B, do not contribute to the structure of water-soluble proteins. It's A. Do you guys think it's A? Right, but we'll see right now. Tamam? C, it has bonding energies of approximately 20 to 40 kilojoules. D, involves the ability of water to denature proteins. Is this possible? And E, primary, involve the effect of polar solutes on the entropy of aqueous systems. But yeah, there's some weird stuff here, but we're going to filter them. So first of all, what do you guys think the answer is? If you guys think it's A, raise your hand. Okay, there's a huge group here. What about you guys? Not A? Okay. How about B? Who thinks it's B? Raise your hands. No? Okay. C? No? No one? Okay. D? No? All right. E? Okay, there's someone here. Okay. What about people at the back? You guys didn't raise your hands. What do you think the answer is? The answer is A. So you guys were correct. The answer is A. Now, can someone tell me why it's A? I want a reason. You have an answer? You were saying something. How did you say it? Anyone else? No? Yeah?
That doesn't react with water. So this formation, this orientation.
Correct. She's correct. Give her a clap, everyone. Okay. So we're going to get to the answer of A in a while. Why is it not B? They do. Exactly. Not is incorrect. Okay. What about C? You can eliminate C and D because you guys didn't really do energy and it's not really related. They're weak. Exactly. So it's not going to be 20 to 40. It will be like 1 to 5. Okay. And as for D, water doesn't denature proteins. In fact, it protects them. As we said, some of the stuff that do denature proteins is pH or temperature when they're in like very high numbers, okay? So as you can see over here, as we know, amphipathic molecules have a hydrophilic head and a hydrophobic tail, right? So as we know, hydrophilic means water-loving and hydrophobic means water-hating. Now, how does this help us? Basically, this is very important for the the structures to form inside cells. And as you can see over here, okay, so as you know, basically what this means is that the hydrophobic tails hate water. So what they're going to try to do is to cluster together and to make a group and like prevent all the water from touching them, okay? So they form a group in the middle of water where they're protected and they can't touch water. And this is known as something called a class trait, okay? And what this is like, it comes from the Latin root clastratus, okay? Which basically means to form a bar or a lattice. So it's basically just kind of like isolated from the water in like its own room, because that's what water does, okay? If you guys want a tip, whenever you're studying and you find a weird word, search for the roots of it, because that can help you understand what this word means, okay? What it helps you with. As you can see, this is important in the lipid bilayer for the protein, not for the protein, for the cell membrane. This is important in forming the lipid bilayer as it forms like, a semi-permeable membrane, okay? So now we're moving on to osmotic pressure. So can someone tell me the definition of osmosis? Everyone say it. What is it? Diffusion of water. Okay. So osmosis is diffusion of water from an area of high water potential to an area of low water potential through a semi-permeable membrane and down the concentration gradient. Now, does osmosis use energy? Passive. Exactly. It does not use energy. Okay. So we've got this question. It's easy, and then we're going to move progress. Do you have a question? Oh, you're already answering. See. Okay, let's take a second. Everyone read the question and analyze it, and then we're going to see what everyone thinks. So the diagram represents two liquids separated by a membrane through which osmosis can occur, okay? So we have a semi-permeable membrane in the middle. What movement of molecules will occur? If you guys think it's A, raise your hands. B, C, okay. Most of you are saying it's C. How about D? Okay, the answer is C. You guys are correct. So can someone explain to me why? Why is it C? Okay, there's a lot of hands. Someone at the back. Add A right there. Can you guys hear? Exactly, correct. Good job. Okay, so as you can see, there's a high solute concentration on the right, and in order to balance both concentrations on either side, the water is going to move to balance the solute concentration, okay? And only water can move, right? Because it's a semi-permeable membrane. It doesn't allow for large molecules like, for example, sugar molecules to pass. Is that clear, everyone? Okay? All right. So a quick revision. As you know, we have three types of solutions. We have isotonic, which means, what does that mean? What is the solution? How is the concentration? Is it high, low, or the same? Exactly, correct. It's equal. So it's the same as the red blood cells, and that does not change the cell volume. Okay, so that's kind of like your red blood cells traveling in your plasma. They're the same concentration. It doesn't change, okay? Now, for the second thing, we have hypertonic solution. What does that mean? Less solutes. Low. Exactly. Hyper means low and tonic means solute. So there's low solute in the concentration. In the solution. Now, what does that cause? It causes cell rupture. Why? Water enters in the membrane. Anything else here? Basically. So water is going to enter from, there's a lot of water in the solution. As we know, water moves from a high concentration to a low concentration, right? So water is going to move from solution into the cell, and that's going to cause the cell to burst, okay? Because it can't take more volume of water than it can already carry, right? And last but not least, we have a hypertonic solution, okay? So does that mean the solutes are more? Exactly, they're high. Okay, so that means that it causes cell shrinkage because there's less solutes outside of the cell, so the water is going to move from the cell into the solution, right? Is that correct? Okay? Okay. So can someone tell me which one represents which solution? I need 3 volunteers, and we're going to... Okay, we have 1. Hamza. Okay. What's the first one? Which solution is this? It shrinks. So what is this? Hypertonic, correct. I should have hidden the answers. It's hypertonic, correct. Okay. Guys, give him a round of applause. Okay, good job. Now, how about the one on the right? What is this? Hypertonic? Correct, it is. That's correct. Good job, whoever answered. Okay, and now we have the middle, which is obviously isotonic because the cells are normal, okay? Now we have 2 more questions before I let you all go. Okay, this is just explaining the transfer of water between the cells and the solution. Okay, so for this question, I want you all to read that, and then we're gonna, again, see what the answer is. The diagram shows apparatus which can be used to demonstrate osmosis, okay? After 1 hour, what would happen to the liquid levels in the glass tubes? So just in case whoever on the back can't see, these are 2 tubes. This one has a highly concentrated solution. This one has a concentrated sucrose solution, and they're both immersed in water with a hole, okay? So water can enter if it wants to. So everyone give it a think, and then we're gonna discuss answers, okay? All right, so if you think the answer is A, put your hands up. What about B? Wait, do you guys want more time? Are we good? A bit more time? Right, okay. Yeah. Are you guys ready? Like a man شوي. Okay. خلاص. Go for it. We're gonna see now. Okay. So if you guys think the answer is A, put your hands up. No? Nobody? All right. How about B? You think it's B? You think it's B? Okay. We're going to discuss now. Why do you think it's B?
Usually osmosis moves from high concentration to low concentration.
Okay, give it a think and we'll get back to you, okay? How about C? Who thinks it's C? Okay, there's more people that think it's C. All right. And last but not least, who thinks it's D? Okay. So I think D is winning. C is a bit tied. Now the answer is actually D. Okay? So who can tell me why it's D? Someone? Anyone? For a new person? Guys, Ismail, let's see. Why do you think it's D? Guys, let's listen to your friends. Exactly. So basically, it's the basis of osmosis. Water is going to move from an area of high water potential. Okay, this is pure water, and it's going to move into an area of low water potential, which is both tubes. Now they're both concentrated, so they don't have much water. So the water is going to move into both of them. So when the water moves in, both of them are going to rise. Okay, they're both going to go up, and that's the answer. Is it clear? Does everyone understand? Anyone have any questions? Anyone? Because we're going to move on to the last question, which is like a challenge. Okay, are you guys good to go? All right, yalla. Bismillah. Okay, I'm going to give you guys a minute. Okay, the diagram shows a plant cell after it has been submerged in solution P for 20 minutes. Okay, so which row describes the water potential of solution P at the start of the experiment and the condition of the cell after 20 minutes? All right. Just in case you guys don't know, turgid means the cell is tight, plasmalized means the cell has shrunk, and flaccid means the cell is weak. Okay, it's like floppy. All right. We'll see. Okay. Give it a think, discuss with your friends, and now we're going to see the answer. Okay, some people are ready. You guys need a bit more time? A bit more time? Okay. Yeah. Okay. Yeah. Okay. Are you guys ready to discuss? By the way, keep in mind, this is like a challenge question, so questions aren't really that hard. Okay, so this is just for fun, all right? Tamam. Okay, let's start discussing. Okay? Are you guys ready? Tamam. If you guys think the answer is A, put your hands up. No one? Okay. If you guys think the answer is B, put your hand up. Some hands here, some hands there. Okay. If you guys think the answer is C, okay, the majority, all right. And who thinks the answer is D? No one. Okay. So is the answer B or C? What do you guys think? C? Yes, the answer is C. Good job for answering. And if you didn't, we're going to explain now, Adi. So why do you guys think it's, whoever thought it was B, why did you think it was B? Anyone? You guys all change to C? Okay, so it's not B. Okay, let's just explain the question. Where is it saying? The question, why isn't it B? Okay, well, not B. Okay, we'll get to that now. So what the question is saying is that basically this plant cell has been submerged in a solution for 20 minutes, okay? And they're telling us that this is the state of the cell after it was in the solution, all right? So we need to know what the solution was before the cell was in it, okay? So they're saying that, as you can see, the cell has shrunk. So what does it mean when the cell has shrunken? What is the solution? Hypertonic? Okay. So if it was in a hypertonic solution, that means that the water potential was high or low compared to the cell. Hypertonic means there's a lot of solutes outside the cell in this area. So how is the water high or low? Low. It's low, right? So if the water is low, water is going to move from an area of high concentration to low concentration of water, right? So where is the higher water concentration? Is the water inside the cell, or where's the water higher? In the cell or in the hypertonic solution? In the cell. So it's going to be moving from inside the cell to the outside, right? Is that correct? And then we're saying that the cell is plasmalized and flaccid because it has shrunken. So when it shrinks, it has no water, it loses its rigidity and it becomes weak, okay? Is it clear? Do you know why it's not B now? Okay? Okay, is this clear for everyone? Tamam? Does anyone have any question about osmosis, about hydrophobic interactions, whatever? Anyone have any questions? All right, that's it? All right. Thank you, everyone. Have a lovely day. And best of luck. Tamam. If you guys have any questions or if you see us around, please don't hesitate to come say hi, come ask a question, okay? We're here to help you and support you. Well, good luck with the rest. Now we have with yes.
Hello everyone, can you guys hear me? All right, thank you guys. So I'm Yasser, and my part is gonna be about living cells. So this is chapter 2, and the way I'm gonna be doing my review is that we will be having basically sort of like the same thing that Jana did. So I'll be asking a question before explaining the topic, and I'm gonna call someone up. And after that, what happens is that if he answers the question correctly, I'm gonna give him or her one of these jelly beans, basically. So yeah. So I want to start with 2.1, which is just basic principles of cells. And these are very important because later on when you guys go into year 1, oh, does it not work here? 1 second. Is it shut off on the sides? 1 second. All right, either way, can you guys see the slide here at least? All right, perfect. So these are very important principles. Why? Because when you turn into year 1 and when you take chapters even later on in this year, these ideas will stack, and you guys are going to encounter these same terms you're going to see in this chapter later on. So many of the things that you've heard, like chaperones, everyone knows here what chaperones are. So they're important in protein folding. So you guys are going to take all of them later on, and hopefully this is just like an introductory session so that you guys, or introductory chapter so that you guys can later on understand their function in more details. So I want to start with the core concepts, which is just water, biological membrane, self-assembly, crowding, molecular machines, proteostasis, and signal transduction. So the first question is going to be about water. So this was covered by my colleagues who came here to do the previous chapter, chapter 3. So what do you guys think is the most, I want you guys to raise hands. What is the most significant interaction that happens between hydrophilic molecules and water? So raise hands. All right, Jihad. Come here. All right, so what is it? What do you think the answer is?
Between hydrophilic and water, hydrogen bonds.
Hydrogen bonds?
Yes.
All right, that's correct. Round of applause. Yes. Yes. So it is hydrogen bonds. You want to straight on? All right, thank you. So it is hydrogen bonds. So before I want to get into why is it hydrogen bonds, I want to break down a number of molecules that are found inside the cell. So this is very basic knowledge. You have first hydrophilic molecules and you have hydrophobic molecules. So hydrophilic molecules are just those that have a lot of electronegative atoms. So like nitrogen, like oxygen, sulfur, all of these, what they do is that they interact with water, they attract water, they like to be with water, and hence the term hydrophilic. Philic means loves. So you can be like, there's not many appropriate terms for this, but so hydrophilic in the sense, like philic means like, and hydro is just water, so it likes water. And when you put those in water, they tend to just interact and bond, and there's no problem. So you can have sugars, for example, you can have salts, sodium, glucose, all of that. And then you have hydrophobic. So you could be, in this case, you have molecules that don't like to interact with water, and these are non-polar in the sense. So why is that? It's because they don't have electronegative atoms that are usually the ones that interact with water. So you'd find most of these are composed of carbon, hydrogen, these kind of atoms. And when you put them in water, they would form clusters. So what do I mean by clusters? Clusters are just like missiles in a sense, like circles, and they would try to stay together as circles and to shield themselves from the water. So you guys know when you put oil in water, it forms sort of like droplets on the surface, right? It doesn't mix, you don't have any sort of anything, you just have droplets in the center. And that's because oil essentially is composed of hydrophobic molecules that form clusters, which are these droplets that stay within the water. So that's about water. And then you have the second part, so I'm going to be asking you guys about cell membrane now. So what happens, or in the sense of the transmembrane domain of any integral receptor. So what does it contain, or what should it contain? So this is related to the part. All right, come on up. Oh, okay. Is it D, phosphate group? No, it does not contain a phosphate group, sadly. Anyone has any ideas? Come on up. Yes, come on up. What do you think the answer is? Oh, first of all, what's your name? Ahmad? All right, what do you think the answer is?
C, polar amino acids.
C, polar, the transmembrane domain. That's not correct. That's not correct, sadly. Non-polar, exactly. Good job. Hand of applause, everyone. Ahmad, your thing. Yes. All right, so that is true. It is non-polar amino acids, and we'll get to that in a second. So you guys all know what the cell membrane is made of. So can everyone tell me in unison what is it made of? A phospholipid bilayer, exactly. And the phospholipid bilayer, the phospholipid itself is a very interesting molecule. And why is that? Is because it's amphipathic. Does anyone know here what amphipathic means? What does it mean? Exactly, that's correct. So it's an amphipathic molecule composed of polar and non-polar parts. So the polar part is the phosphate head, which has a phosphate group. So it has oxygen, phosphorus, all those electronegative atoms. And you have a hydrophobic fatty acid tail full of carbon of hydrogen that does not like to interact with water. So this structure, this amphipathic nature, is what allows it to be selective in a sense. So what do I mean by selective? When I say selective, I mean I allow certain things in, and I only allow these things in. And pardon? Yes, exactly. So it is semi-permeable or it's partially permeable. So in that sense, what does that mean? Is that I only allow certain things in, and the rest I do not. So I select. It's like border control in a sense. They only allow some nationalities in, and others they just keep at the border, sadly. So yeah. So you can think of it in that sense. So that's what cell membrane is, and you're going to see this recurring a lot every time you take anything regarding receptors or cell membranes or anything like that. So you're going to have integral proteins, which are the proteins that pass through the whole membrane. So they go from the inside and the outside. And with those proteins, you have a transmembrane domain. So what does transmembrane domain mean? It means that it passes through the whole thing. So on the parts that align with the polar phosphate group, you're going to have polar amino acids because you have to match your energy in a sense. They have to match each other's energy. So hydrophilic molecules or the hydrophilic amino acids are only going to be the ones that are next to the polar phosphate group. And in the middle, where you have hydrophobic, there's going to be the hydrophobic non-polar parts. And hence the answer to this question. So the non-polar amino acids are going to be the hydrophobic ones, which are going to be next to the fatty acid tail, which is hydrophobic, right? So you guys understand what I mean? All right, perfect. And you have peripheral proteins, which are the ones that are only found on one side. So they can be found on the inside, found on the outside, and they only are like on the surface in a sense, or even partially embedded, they're only on the surface, and they move around. Everything moves around. The cell membrane itself is a very dynamic structure. So don't think of it like a rigid cell, like it's just a block, like a Rubik's cube that doesn't move. It's more of like an escalator or like a big crowd rushing in, and you have everything that moves. You know what I mean? So you're gonna take this later on. It's called the fluid mosaic model, if you guys are familiar with that. So they all move laterally and they vibrate and everything like that. So this is for cell membrane. Moving on to the next concept, which is self-assembly. So what type of interactions primarily hold the supramolecular complexes together during self-assembly? I want new hands. Anyone has any idea? Go ahead. Yes, correct. Here you go. Round of applause, everyone. So that's correct. They are held together by non-covalent interactions. And before I get to self-assembly itself, I want to explain to you guys what a supramolecular complex is. Because personally, when I read that phrase, I was like, what is that? So supramolecular complexes are just proteins that have multiple subunits. They're composed of multiple parts. And these parts, we call them self-assembling. So what does that mean? You can think of each subunit of the supramolecular complex as like a strand of hair in a sense. So you know how some people have curly hair or kinky hair or anything like that, right? So what that means basically is that no matter how much you try to like stretch it or move it or change it, it always tends to go back to a certain way, right? Or to a certain shape. So this is similar to these complexes in a sense because these complexes, they're composed of proteins that bend and fold in a certain way that just happens to be complementary to each other. So an example of this could be ribosomes. So ribosomes, you know, are composed of multiple things like proteins and RNA, and all of these fold in a way that don't require energy, and they fold in a way that allows them to just it's like folds in a way that allows them to be complementary to each other. And they're held together, as she said, by non-covalent interactions. So like hydrogen bonds, hydrophobic interactions, ionic bonds. So all of these things are what's holding the proteins together. And you have certain conditions. Obviously, you can't have this happen all the time. There are certain, you can say like, abnormal conditions in which you have to regulate it. And in that case, you can use chaperones. So like we said, they help in protein folding and getting everything together. And the other thing is that you have to have a sort of balance between them. So obviously, if between different molecules, the hydrophobic parts will interact with the hydrophobic parts and hydrophilic parts will interact with the hydrophilic parts. So you guys understand, this is a very important principle. You guys have to understand it. So you guys understand what I mean? Yes.
Yes.
All right, perfect. Thank you. All right, moving on to the next question, we're going to move on to biological machines or molecular machines. So why are molecular machines considered efficient in their use of energy? Anyone wants to say anything? What's your name? Sarah. All right, you want to come up? You don't want to come up? All right, that's okay. What's your answer? B? Yes, that's correct. Good job. Round of applause, everyone. So yes, it's very correct that it is the hydrolysis of ATP being irreversible. That's what causes the molecular machines to be very efficient. Because when you have an irreversible reaction, obviously, and specifically for the hydrolysis of ATP, because that's meant to release energy. So when you release energy and you make that irreversible, you will have a more efficient reaction in a sense. Do you guys understand what I mean? So now we're going to move on to the molecular machines itself and what their whole cycle is. So molecular machines are similar to a lot of supramolecular complexes. They can fall under the you can say molecular machines. Sorry, they fall under the category of supramolecular complexes because they're just proteins that are composed of different subunits, multiple like parts, and these just happen to move. So an example that I've mentioned in the previous slide was ribosomes, and this is also consistent with this slide. So molecular machines, ribosomes are considered molecular machines because what happens with molecular machines is that ATP or GTP, they bind to the machine, they bind to one of the subunits. The subunits will hydrolyze this ATP and release energy. And then this energy is used to do what? Its energy is used to change the shape of this one subunit, and then like dominoes falling, every subunit around it will also change in shape. And that causes movement in one direction only. So one of the options previously was that it moves in both directions, and that's wrong. It only moves in one direction, all right? So that change occurs in one direction only, and yeah, that's basically it. Be mindful of the examples, because they could give you like an example, like I mentioned, ribosomes, and they could tell you that it moves in one direction, and about the whole and they could be asking you like, is this considered a molecular machine or something like that? They could give you not like out of the book examples and asking and giving you characteristics of a molecular machine, and then you have to decide based on this, is this a molecular machine or is this not? If they tell you, is it moves in one direction, is it energy independent? So in both of these cases, if it does not use energy, it's not a molecular machine, and if it does not, and if it moves in more than one direction, it's also not a molecular machine. So you guys understand what I mean? All right. And then we have the proteostasis. So proteostasis is, let's just start with this question actually. So what is the function of molecular chaperones and proteostasis network? Anyone here know? What do you think? Yes, you. C? It is to assist in protein folding and unfolding. That's correct. Good job. Round of applause.
Hello everyone, how's everyone doing? We had a slight issue with the audio from this point of the PACademia lecture till the anterograde part of the Golgi body, and so I've decided to re-record it from the comfort of my own dorm. I hope you guys find this helpful, and I'm sorry for the disturbance. Alright, so in proteostasis, what happens essentially is that it is quite literally, the word proteostasis quite literally translates to protein, and the word stasis means regulation, or means control, or means to freeze. So everyone knows obviously that we carry out protein synthesis in our own cell, and sometimes we have misfolding in some proteins, and we have problems with the patterns and the modifications of proteins. And when you have misfolded proteins, these are not good for the cell because their accumulation can result in disturbances in the reactions inside the cell. So it's obviously not good. And this accumulation is what we call proteotoxic stress. And it can happen from temperatures, from oxidative stress, from genetic variations, from mutations, all of that. So obviously this is not a good thing, and the cell does its best to control the accumulation of these misfolded proteins by the process of proteostasis, and it does that through molecular chaperones, through enzymes, and through other pathways. So the molecular chaperones are mostly involved in making sure the protein does not misfold in the first place, as you guys are going to take in the chapter proteins and enzymes, I believe. And proteolytic enzymes are there to digest and break down the misfolded proteins to prevent their accumulation, and that also happens with lysosomal degradation and autophagy. Moving on, we're gonna now study the 4 stages of signal transduction. So can anyone tell me what they are? I'm gonna give you guys 5 seconds. So the 4 stages of signal transduction are: number 1, you have reception, which is where the ligand binds; number 2, you have transduction; number 3, you have response; and number 4, although it's not mentioned in the slide, you have termination. So signal transduction is a very, very important mechanism. And why is that? It's because most of the signaling molecules inside your body act through signal transduction. So an example would be neurotransmitters, they act through signal transduction, hormones act through signal transduction, cytokines act through signal transduction. So it's a very important mechanism. And how does it happen? We'll see just now. So you have a ligand, could be a hormone like we said, or a neurotransmitter, and it binds to a receptor on the plasma membrane, and then that triggers some sort of conformational change. And this conformational change is what triggers the transduction, because this causes the release of a second messenger. A second messenger is essentially a molecule that is responsible for triggering the enzyme cascade, which is the 3rd step. So what we have first is ligand binding and conformational change, and then this triggers the production of a second messenger, and then you have this second messenger triggering the response. So this response would be, for example, the second messenger would come and cause the phosphorylation of one enzyme, and therefore activating it. And then this enzyme would phosphorylate another enzyme, activating it. And this enzyme would phosphorylate another enzyme. So that's why it's called a cascade, because it's more of like dominoes falling. So this enzyme activates this, and then this, and then this, and this is what produces a response. So this response could be to, for example, produce a large molecule, or it could be to increase permeability of the membrane of the cell to something. So all of these are responses or examples you're going to take. So for the purpose of this slide, I'm going to be using the example of glucagon. So everyone here knows that glucagon is the hormone that's responsible for increasing the blood sugar. So the hormone that is glucagon here acts as the ligand, and it binds to the receptor, and it triggers a conformational change in the receptor, which causes it to dissociate. You don't have to know all of these details, this is just an example for your understanding, which causes it to dissociate. And then this releases a second messenger that's called cyclic AMP, and then this second messenger triggers the activation of different protein kinases and different enzymes, and the response here is what? The response here is to break down glycogen. The response here is to increase the membrane's permeability to glucose by adding glucose channels so that the glucose can pass outside the cell. So this is just an example, you guys don't have to know. Another important detail is that calcium ions are a very common second messenger, and they're especially important in muscle cells and nerve cells, and you guys are going to learn that later on. So this is just for your reference. So now we will be moving on to prokaryotic cells and all their different components. So let's start with the cell wall. So this question says, E. coli is known as a gram-negative bacterial species. So the first question is that how is it determined? How is it determined whether a bacteria is gram-negative and gram-positive? And how do gram-negative bacteria differ structurally from gram-positive bacteria? So the answer to this is that the determination of whether the bacteria is gram-negative and gram-positive is determined scientifically by whether the bacterial cell wall absorbs a stain called the gram stain. So gram-negative bacteria would not absorb as much of the stain, and that's why they appear pink under the microscope, while gram-positive absorb a lot of it, and that's why they appear purple. The colors don't really matter, you guys just have to know that there's different, gram-positive bacteria absorb the stain a lot more and a lot better than gram-negative bacteria. And how do gram-negative bacteria differ structurally from gram-positive bacteria? So the reason why gram-negative bacteria in the first place absorb less of the stain is because they have a thinner cell wall, and this cell wall is made up of a peptidoglycan layer, a peptidoglycan polymeric network, which just means that multiple peptidoglycan units are just linked together by covalent bonds. So it has a thin layer, and most of the cell wall is more of a fluid cavity, while in the gram-positive bacteria, there's a very thick layer of peptidoglycan, and that's why it absorbs the stain better. And here, which part of the bacteria becomes an endotoxin? So it's very important to note that this endotoxin ability is exclusive to gram-negative bacteria, and this is because the answer here is D. It's lipopolysaccharide. So we're going to see in a bit why that is correct, as we'll see the differences between gram-positive and gram-negative bacteria. So just keep in mind that the endotoxin is produced or it's caused by lipopolysaccharide. And then you have, last but not least, which one of these is not a function of proteins that are found inside a gelatinous layer? So I want you guys to think about what this gelatinous layer is. So we're going to see that in a bit. This gelatinous layer is the periplasmic cavity, or the periplasmic space. This periplasmic space contains a lot of proteins, and their functions include chemotaxis, transport, and enzyme activity, but it does not include receptor binding. So here, receptor binding is the correct answer because it's not a function of the periplasmic space. So let's look at the differences between bacterial cell walls and the different types of bacterial cell walls. So here you have gram-negative and gram-positive. This is actually pretty interesting because I learned about bacterial cell walls this week in our microbiology lecture. So what this means is that this is very important information that you guys are going to keep taking later on with more detail. So it's very important that you guys focus on this, especially since it's important portions in year 1. So for the gram-positive bacterial cell wall, as you see, you have a very thick layer of this green material. So this green material it's not actually green in real life, this is just a diagram, but this thick layer is the peptidoglycan layer. And because of how thick it is, it absorbs more of the gram stain, and that's why it appears as a darker color under the microscope. While we have on gram-negative bacteria, we have a thinner layer of peptidoglycan, and instead you have an outer membrane. So it's important to note that the gram-positive have only one membrane, while the gram-negative have 2 membranes. And the outer membranes is very important because it is what contains lipopolysaccharides. So this is different from a normal cell membrane because normal cell membranes are made of phospholipids, right? But this outer membrane is made up of something called a lipopolysaccharide, which is composed of 2 things. It's composed of lipid A and just a polysaccharide. And this lipid A is what causes symptoms to arise, because when your body breaks down this bacteria, the lipopolysaccharide is broken down and lipid A is released into the blood, and this lipid A is what becomes an endotoxin and causes you to have symptoms like fever and all of that. And also you have the periplasmic space, which, as we mentioned, has proteins that are responsible for chemotaxis, transport, and enzymatic activity. It's also important to note that there are channel proteins that are found on the outer membrane, which help with the transport. So all of these are the differences between 2 of them. So this diagram is just another illustration. A very important detail is the glycocalyx, and the glycocalyx is quite literally the word glycocalyx. It means sugar coating. So this glycocalyx serves 2 functions in bacteria. So if it is organized, we call it a capsule, and capsules are a huge physiological advantage for the bacteria. It's a huge advantage for bacteria because it helps them evade phagocytosis. It helps them evade immune cells, and it helps them evade the immune system and the destruction that the immune system brings on these bacteria. So it protects them from being destroyed by the immune system, so that they can linger in your body further. So that they can stay in your body and feed off of it and multiply and replicate and cause more damage. This is if the glycocalyx is organized. If it is not organized, if it doesn't have a pattern, and if it's loose, we call it a biofilm, or we call it a slime layer. So in this case, the purpose of the glycocalyx is just to stick, to adhese. It's more of an adhesive surface. It helps stick to surfaces, like for example, stick surfaces like your teeth, and it helps to stick to surfaces and just produce, and over time it produces more and more sugar and carbohydrates as it sticks to those surfaces. And this is actually what causes tooth cavities, because what happens is that this bacteria settles on your teeth and uses the glycocalyx to stick to them, and over time they produce more and more carbohydrates, and these carbohydrates and these enzymes start to degrade your teeth and start to cause cavities. So this is what the purpose of the biofilm is. Moving on, now we have the cell membrane. So as I mentioned previously, the cell membrane is just a very recurring theme, as you'll see in all organisms, and it always has a similar function. So we've already mentioned that cell membranes are made of a phospholipid bilayer. They have integral and peripheral proteins. The peripheral proteins are bonded to phospholipids covalently. And you also have receptor proteins that are found on their surface, and transport proteins, which are the transmembrane proteins we've talked about. There are two unique features about prokaryotic cell membranes, is that they have hopanoids or hopanoids. These hopanoids, they stiffen the membrane. What do I mean by stiffen the membrane? Is that when they put themselves between two phospholipid molecules in order to make things more crowded. And as the phospholipid or the cell surface membrane becomes more crowded, the membrane becomes less permeable. So this is an advantage if the cell surface membrane wants to, or if the cell wants to regulate more, or like to control more, or to like kind of have more control over what goes inside the cell and restrict the movement of substances. So yeah, and then you have more proteins that are found on the surface of the surface membrane on prokaryotes, and this one is very important. So as we already know, the prokaryotic cells, they don't have any membrane-bound organelles. And what I mean by that is that they don't have chloroplast or mitochondria, and these are very important for energy generation, right? So how can the prokaryotic cells produce energy if they don't have mitochondria? So as an adaptation, or to cope with that, the prokaryotes have proteins on the cell surface membrane that carry out photosynthesis and respiration and produce energy for obviously the metabolic processes inside the cell. So this is another question here. Which of the following best describes plasmids? Is it required for growth and cell division? Does it contain the majority of the cell's genetic information? Does it replicate independently of the chromosome, or is it made up of large linear molecules? So let's go through these one by one. The plasmids are as we know, are something called extra chromosomal. What do I mean by the word extra chromosomal? Is that they are extra chromosomes, not chromosomes. They're extra genetic material, and thus the cell doesn't need them, but the cells like to have them because they give the cells a lot of unique advantages that helps them survive. So survival is not necessarily, the bacterial cell can survive without plasmids, but if they have them, they'll be more likely to survive under more harsh circumstances. So do they contain the majority of the cell's genetic information? No, that's just the nucleoid, as we'll see in a bit. Are they made up of large linear molecules? No, they're made up of large circular molecules. So the correct options are yes, they do replicate independently of the chromosome, which is B. So this is another question here. Which inclusion bodies assist in phospholipid synthesis? So phospholipid synthesis obviously is going to be polyphosphate granules. It's quite literally in the name. But let's go through what each one does. So let's start with A. Glycogen granules obviously are for energy generation or for energy storage. Magnetosomes are unique because they are used by the bacterial cell for navigation. The cell, in a weird way, aligns itself with the Earth magnetic system, and then it uses that for navigation. You don't have to know all of that. It's just for general information, general knowledge. They might ask like a small thing about it, but don't worry about it. And sulfur granules also have the same purpose as glycogen granules. In some bacteria, sulfur and sulfur compounds are oxidized instead of carbohydrates in order to give you energy. So they're not the answer. The only answer left is polyphosphate granules, which assist in DNA synthesis and phospholipid synthesis. So now let's look at the prokaryotic cytoplasm. Let's start with the genetic information. So obviously we've already mentioned that there is no nucleus inside the prokaryotic cells, and therefore you just have genetic information that is coiled, that is super coiled in some regions and super loose in other regions. And the place where you have this genetic information is called a nucleoid. So this is very important. And it's also important to know that the whole everything here, it's labeled as the chromosome in the book. So it's not exactly a chromosome in the conventional sense, because in eukaryotic cells we have chromosomes. We as humans, we have chromosomes, and they're made up of DNA wound around histone proteins. So prokaryotic cells don't have histone proteins. So this is just labeled as the chromosome because it's just genetic information that's found in the middle of the cell. And this is for the DNA, and the genetic information, let's talk about the inclusion bodies. So the inclusion bodies are just kind of like granules or like specks that are found inside the cytoplasm. And as we've mentioned, we have 4 types: glycogen granules for energy, polyphosphate granules for phospholipid and DNA synthesis, sulfur granules for oxidizing sulfur compounds for energy, and magnetosomes are for navigation. Yes, and then now we have the last part of the chapter, and probably the biggest part, which is eukaryotic cells. So, as a bit of a background, it's important to note that there's a huge difference between obviously eukaryotic cells and prokaryotic cells. Eukaryotic cells are much larger, and they have membrane-bound organelles, and they have a nucleus and all of that. And another important thing is that they contain something called an endomembrane system. Now, what is an endomembrane system? An endomembrane system is essentially just similar to an organ system, but it's an organelle system. So a bunch of organelles that are found inside the cell, they work together to form the endomembrane system. And their function mostly is the fact that they are responsible for the production, the modification, and the transport or the export of proteins or molecules generally from the inside of the cell to the outside of the cell. And they are connected either by membranes directly, so they connect to each other by membranes, or they're connected by vesicles. And I'll also see that in a bit. So the organelles that are part of the endomembrane system include the nucleus, the endoplasmic reticulum, the Golgi body, the lysosomes, and finally the plasma membrane. And they all have an important role. So yes. So what are vesicles? So vesicles are just like bubbles containing molecules inside of them, and their job is to transport molecules from a donor membrane to a target membrane. So donor membrane is where stuff is produced and it's packaged and released, and target membrane is where it's received. And their movement around the cell is assisted by obviously coats of proteins. This is not very important. You just have to know it as like information or just knowledge. So as you see here, this is the endomembrane system. You have a nucleus, and it's connected by a membrane to the rough endoplasmic reticulum. This is the membrane right here, and it's also the rough endoplasmic reticulum is connected to the smooth endoplasmic reticulum by membrane as well. And then we get to the Golgi body. The Golgi body is not exactly connected. It's not connected at all by a membrane to the rough endoplasmic reticulum or the nucleus. So this is where vesicles come into play. So these vesicles, as we'll see in a bit, these vesicles, they bud off from the endoplasmic reticulum and fuse with the Golgi body. And in the same way, these vesicles bud off later from the Golgi body to the plasma membrane. So this is how the endomembrane system is connected. Before we dive deeper into the endomembrane system, I want to talk about the cell surface membrane of the eukaryotes. So yes, so this is a question here: which one is not a function of the eukaryotic glycocalyx? So are they there? So let's see. Are they used for self-recognition? Yes. Are they used for receptor binding? Yes. Are they used for titer binding with other cells? Yes. The only thing they're not used for is to resist mechanical stress. So C is the correct answer. It's not a function of the eukaryotic glycocalyx. So here, I remember I made a mistake in this question in the actual lecture. Which is true about the membrane skeleton? Is it made up of myosin? Does it protect the membrane? Is it extracellular, or is it made of actin? So let's go through them. Is it made of myosin? No, it is not made of myosin. Does it protect the membrane? It does not protect the membrane. It doesn't really protect the membrane. Its purpose is more for reinforcement. It's not extracellular. It's found inside the cell. And last but not least, and therefore it only leaves actin. So yes, the membrane skeleton is made up of actin. So be careful not to confuse yourself between this and the molecular machines I mentioned earlier, because I'm not sure if I mentioned this, but actin and myosin together work to produce molecular machines. So here it's just actin forming the membrane skeleton. Yes, alright, perfect. So this is the cell membrane, and it's the same thing as the functions are similar for the receptors and the peripheral and integral proteins in the prokaryotic cell. The difference I want to highlight here is the glycocalyx. So the function of the glycocalyx in the prokaryotes was mostly for protection, for evasion. Here in the cell membrane of eukaryotes, it's mostly for identification, for recognition. So you could think of the glycocalyx like a carbohydrate fingerprint. So you know how fingerprints are used for identification in real life? The same way, your body and your immune system uses the glycocalyx to identify which cells belong to this body and which cells do not belong to the body. And this is also significant in cancer, for example, because when the cell turns cancerous, the glycocalyx changes, and this sometimes would help the immune system detect, okay, this cell, it has a different glycocalyx, we don't recognize this glycocalyx, so this cell is not our cell, there's something wrong with it, so let's destroy it. So sometimes this would alert the immune system to cancer cells, but cancer cells are very adaptive and they change their glycocalyx very often, so the immune system has a really hard time picking up on them. We digressed, but let's go back to the main functions of the glycocalyx. So the main function of glycocalyx includes cell-to-cell recognition. So the cells that are next to each other, they recognize each other by their glycocalyx, and this would help them communicate with each other. And then you have adhesion. So this is essentially also important for when you want very tight binding between the cells. So the glycocalyx helps with that. And you have receptor specificity. What this means is that there are some receptors, there are some carbohydrate molecules that are found on top of the receptors that help the ligands find them and bind to them, so this is important. And obviously self-identity, like we've mentioned, which is for the immune system. Another thing that I want to mention is, as I've talked about, it's the membrane skeleton. So as you see in the diagram here, the membrane skeleton is intracellular, and it's made up of actin filaments, and its purpose is just for reinforcing the membrane, not protection, reinforcement. And you also have another thing that I want to discuss, which is the extracellular matrix. So the extracellular matrix here, you guys have already learned it in tissues. It's just a fluid that has fibers that is produced by fibroblasts, and the purpose of these is for binding cells together, for keeping them in a specific place. And another thing is the most important thing actually is that these fibers and the extracellular matrix generally, it regulates and it controls how the cell talks and how the cell interacts with its environment. So this is also very important. Yes, so now we're moving on to the endoplasmic reticulum. So what is the fate of transmembrane proteins in the rough endoplasmic reticulum? Let's go through them. Does it enter the lumen of the RER? No, this is just for water-soluble proteins. Does it undergo folding? Now, if it cannot enter the lumen of the RER, how can it undergo folding? So this is wrong. And if it cannot enter the lumen of the rough ER, how can it undergo glycosylation either? So that all of these three are wrong, and the answer is that it remains in the ER membrane, and we're going to see why that is in a bit. So yes, moving on to the endoplasmic reticulum. So there's two different types of endoplasmic reticulum, and even though they're connected to each other through a membrane, their difference is huge functionally. So the rough ER is responsible mostly for the processing of proteins, while the smooth ER is responsible for lipid processing and lipid production, along with other functions that we're going to be discussing. So the rough ER is called rough because its surface is rough. And why is its surface rough? It's because it has ribosomes on its surface. The ribosomes coat and they surround the surface of the rough ER. It's very important to keep in mind that it's not the rough ER that produce the proteins, it's the ribosomes on their surface that produce the proteins. What the rough ER does is protein processing, not production, processing. And it processes two different types of proteins. It processes membrane proteins and water-soluble proteins. So as a question asked, what is the fate of membrane proteins? Membrane proteins have a hydrophobic region. So as we've mentioned, every membrane protein, every integral protein has to have a hydrophobic domain or hydrophobic part. So this hydrophobic part, when you produce a protein, when the ribosome produces a protein on the rough endoplasmic reticulum, the protein is usually pushed into the membrane and into the middle of the endoplasmic reticulum, into the lumen. So since the membrane proteins have a hydrophobic domain, as it's going through the membrane of the rough endoplasmic reticulum, this hydrophobic transmembrane domain or transmembrane portion, it aligns with the hydrophobic part of the phospholipid bilayer in the rough endoplasmic reticulum's membrane. I know this sounds like a lot of words, so let me say it again. So proteins produced by ribosome. It's pushed into the lumen, and as it's pushed into the lumen, it has to pass through obviously the membrane of the rough ER. So since the membrane proteins have a hydrophobic part, it aligns and it sticks with the hydrophobic part of the membrane, and therefore it stays in the membrane. So that's for membrane proteins. Water-soluble proteins, obviously, they do not stick with the hydrophobic portion, and therefore they're pushed into the lumen of the rough ER. And the most important modifications generally are protein folding by chaperones in the rough endoplasmic reticulum and glycosylation. Glycosylation is just when you add sugar or carbohydrate molecules to a protein. An important thing here as well is that the rough ER has a big role in proteostasis. So do you remember when we studied proteostasis and how it's management of misfolded proteins? So this is very important here because this is where protein synthesis and processing happens. So this is where all the errors could occur. So initially, the first response to the endoplasmic reticulum, the first response by the rough endoplasmic reticulum when you have proteotoxic stress is that it triggers something called ER-associated protein degradation, which is where the proteins they bud off into a vesicle from the rough ER, and that vesicle fuses with a proteasome. So the proteasome essentially is just a vesicle containing hydrolytic enzymes that break down proteins. They're called proteolytic, and that's why the vesicle is called proteasome, and they fuse each other and the misfolded proteins are broken down. So this is simple and easy. Now, if the proteotoxic stress cannot be managed by the ER-associated protein degradation, then we move to the unfolded protein response. So the unfolded protein response is where you have too much cytotoxic stress, and this not cytotoxic stress, the proteotoxic stress. And what this results in is that the ER sends chemical and signal molecules to the nucleus to stop producing mRNA and stop producing mRNA so that we can stop producing proteins overall, to prevent the process of protein synthesis. So in this case, there's no protein synthesis. So for the cell, it thinks in a way that's like, okay, if there's no proteins being produced, then there cannot be any proteins that can be misfolded, and therefore I can prevent proteotoxic stress. So this is how the cell thinks about it. And then you have another thing, which is autophagy, which is where it's not mentioned in the slide, but it is mentioned in the book, that you have autophagy, which is where the proteins are also eaten up by hydrolytic enzymes and by hydrolytic enzymes. And then you have apoptosis. Apoptosis, where there's too much proteotoxic stress and the cell cannot handle it, and it's affecting cellular function too much, and therefore the cell decides to kill itself, or it's called programmed cell death. So it starts to just kill itself essentially. Yes. So yeah, that's for the rough ER, and then you have the smooth ER. The smooth ER is not very significant. It's very important actually. It's just that there's not much explanation to go over. You have production of lipids and steroids, and they're also very important in muscles where they store calcium ions. When you guys take the muscle chapter in Bio 1, you're going to see the importance of the smooth endoplasmic reticulum. In muscles, it's called sarcoplasmic reticulum, and it's also responsible for biotransformation, which is where, especially in the liver, it converts molecules inside the body, especially toxic molecules, into water-soluble molecules that can be excreted, that can be removed from the body. So yeah. And then we have which structure of the Golgi apparatus is primarily responsible for receiving newly synthesized proteins and lipids. So this is very obvious. It's this phase. The transphase doesn't receive it exports, and we'll see the other structures in the next slide as well. So now this is a very important portion. I want you guys to focus. So you have three different types. So you have three different steps in the function of the Golgi body. So the first thing that happens, let's start with anterior grade transport, which is where the molecules, this is before they arrive to the Golgi body. This is as they are arriving to the Golgi body. So after, for example, a protein or a lipid has been produced in the endoplasmic reticulum, it's released in a vesicle, and that vesicle moves along microtubules towards the Golgi body. And as they reach the Golgi body, these vesicles that contain different proteins and different lipids, they fuse with each other, and the vesicle fusion, it creates a big creates something called a vesicular tubule cluster. So it's a lot of vesicles next to each other, the vesicular tubule structure. And then what you have is that this VTC, it forms the cis face of the Golgi body. So obviously now we've created a lot of vesicles fusing each other, so it forms a large cistern or a cisternae, and that becomes the cis face of the Golgi body. And then you have retrograde transport. Retrograde transport is basically the idea that, the vesicles that we use to ship off, to send the proteins from the endoplasmic reticulum, they were containing endoplasmic reticulum proteins. So why would we keep those proteins? So now that they're fused together and now that they form the cis face of the Golgi body, why do we want to keep them there? We don't want to keep them there. Let's recycle them and send them back to where they function, which is the endoplasmic reticulum. So this is what retrograde transport is that we are budding vesicles containing ER proteins from the VTC and returning them to the ER. So yeah. And then you have the cisternal maturation model, which I believe I've explained in person, and yes.
Vesicles that contain the rough ER proteins, they're recycled back to the rough ER. So that was anterograde because it moved to the Golgi body, and that was retrograde, retro meaning like going back because it moved back. And then you have the cisternal maturation model, which is not really a step, it's just how the Golgi body works, is that as more VTCs come, they form the cis phase. And the cis phase moves down as more vesicles come and becomes eventually a trans phase. So that's basically how it works. And while the cis phase is moving forwards and downwards, protein modification happens. You have glycosylation, you have phosphorylation, you have sulfation, all of these things. So yeah. And then you have endocytosis. So this is not really important. I think the doctors mentioned that you guys only have to know the figures, right? So all you guys have to know is that the molecule binds to a receptor on the plasma membrane, and then it moves, and it moves inside. And the receptors that are found in the vesicles are they are recycled back to the plasma membrane, and this vesicle has one of three fates. It either fuses with an early endosome, or what happens is that it goes to the Golgi body, or then you can have proteins from proteins from lysosomes called acid hydrolases fuse with it, and it becomes its own lysosome. So this is all you have to know. These are all steps that link each other, but in reality, it has one of three fates. So it either goes back to the cell membrane and it's recycled, or vesicles come from the Golgi body containing those acid hydrolases that fuse with this endosome to form a lysosome, or you have these come to the Golgi body and fuse with the trans phase for further processing. You guys understand? I feel like it's a lot. Go ahead. Yes. The old trans phase, it's okay, perfect question. I wanted to get to that. So the trans phase eventually dissolves and becomes secretory vesicles, because as the cis phase goes from cis to trans, you have all the modification that we wanted to happen. It happened, right? So now it's time for secretion, for exocytosis. So all this trans phase, it dissolves into secretory vesicles, and it goes to the membrane and releases its product. Exactly, basically. So do you understand? All right, does anyone have any other questions? No? All right, perfect. So now this will be getting to towards the end of the chapter almost, is that the nucleus. So the nucleus has a lot of details I've noticed in the book, and a lot of them are very unnecessary and just huge and boring. So I've just tried to summarize my best for this part. So the nucleus is obviously the most important part, or the most important organelle in the cell, and I want to get to the structure first. So the nucleus contains a nucleolus inside, which contains all the important DNA, all the chromosomes, all the chromatin, and it has a nucleoplasm, which is kind of like the cytoplasm, but for the nucleus itself, and it's surrounded by a double membrane. So you have to know that only two organelles in the cell are surrounded by a double membrane, which are the mitochondria and the nucleus. Okay, and these inner membranes and outer membranes, they have different functions. I will be seeing in a bit. And the space between them is called the perinuclear space. So this is just like basic structures. And what you have to know is that there is different types of kind of like genetic material depending on which stage of the cell that you're in. So this is time for a question. So which chromatin would not be affected by a toxin that inhibits transcription? I know it's a lot to take in, but I want you to break down. So which type of chromatin would not be affected by a toxin that inhibits transcription. So what do you think? I want new hands. Does anyone here know? Here, this side in the middle. You can just have a guess if you'd like. You can just have a guess if you'd like. Go ahead. C, constitutive heterochromatin. Yes, that is correct. Good job. Round of applause. Your one. So yes, so we'll be seeing now in a bit why the answer C is correct. So there are two types of chromatin generally in the cell. You have heterochromatin and euchromatin. So heterochromatin is the one that is not. It's the one that is. The heterochromatin is the one that's not used for. It's not used for transcription. It's not transcriptionally active. And why is this? And this is why you tend to find it in the periphery of a chromosome. Because since we don't use it, let's keep it at the end. And then you have euchromatin. Euchromatin is the part that's used for transcription, and you'll find that it's very loose. It's very loose. If it's loose, you can imagine like this. If you have a lot of things packed together, like for example, when you have like a chain and you do a lot of knots with it, it's very hard to pick out one certain piece. But if you loosen it out, it's very easy for an enzyme. It's very easy to like feel every single piece. And this is important for transcription because what is going on is that the RNA polymerase enzyme has to come and feel and go through every single nucleotide to prevent errors. And this cannot happen if you're densely packed. So the euchromatin tends to be in the center of a chromosome, while the hetero tends to be at the periphery. And there are two types of heterochromatin depending on whether they can turn into euchromatin later on or not. So you have the constitutive part, or the constitutive type, which is always turned off. It never turns into chromatin, and that's why it's not affected by transcription, because it never even undergoes transcription. It's always dense. And then you have the facultative euchromatin, which can change. It can go from inactive to active. It can go from dense to uncondensed, depending on the cell's needs. So you guys understand? All right. So I have another question. Which one of the following regulates entry of molecules into the nucleus? So this is to do with the nuclear pore center. What do you think? Yes, correct. Good job. Where did he go? Oh, I'll get it for you. Well, I think you went to the other side. I think you went to the other side, yeah. Oh, yeah, okay. I'll just give this to the next time. Thank you. So yeah, it is nuclear porins. So I want to explain now how this thing happens. So it's a bit of a mess, so just bear with me. So if anyone has any doubt, ask me. What happens here is that this is the nuclear pore, and it's also called as a nuclear pore complex. You guys know the nuclear pore is what? It's the space. It's the space that is in some parts of the nuclear envelope where it's open to the cytoplasm. All right. And here you have a ring or a basket of proteins that have protruding strands. And this is just for docking. So what does docking mean? Docking is like, when a ship docks in like a port, so it stays there. So this is just to prepare these molecules for transport. So these molecules are the ships, and they're sitting there with the help of this basket of proteins, which calls the ring-shaped core, and these large molecules stay there for transport. Now, what decides whether these molecules can go in or out, like she said, is nuclear porins. So these decide whether these molecules that have docked here can go in and out. And only certain molecules can go in or out. You have, for example, RNA or certain proteins, only these are allowed. And the others are not allowed. And if something wants to go out from the nucleus into the cell, then that's helped with what's the name of the protein that makes it go out from inside the nucleus to outside the nucleus? Someone say something? Correct, it is exportin. And if you want to move something inside the cell, inside the nucleus, exactly. So that's correct. So this is everything to do with the nuclear pore center. And yes, obviously this process uses GTP and ATP for energy because it's a very energy consuming process. And you have one more thing, which is the nuclear lamina. The nuclear lamina is lying on the inside, on the inner surface of the membrane, and what it does is just provide support. It provides support, and it has other roles. It can bind chromatin, it can contribute to replication, for transcription, all of that. And that's basically it for the nucleus. Now, coming to the last part of the chapter, which is mitochondrial fusion. So, just generally just mitochondria. So anyone can answer this question? So what is the role of mitochondrial fusion in cells? Go ahead. B. It helps rescue damaged mitochondria. Mitochondrial fusion? Yes, that's correct. Good job. Round of applause. It's okay, I'll give it to her. Oh. She's over there. Sorry. Yes, so it does help rescue damaged mitochondria. So now we'll be getting to the mitochondria. So the mitochondria, generally, you guys have all heard this, it's the powerhouse of the cell, and that's quite literal. It carries out aerobic metabolism. It has a role in other processes as well, as you guys will be seeing in semester 2 when you take biochem with Dr. Wasim. He's going to be explaining the urea cycle, all of the process of aerobic respiration in more details, and that's how these are the processes that the mitochondria contributes to. You guys just have to know them by name by now. So which is just like it helps with metabolism of urea. They produce heme groups. They produce iron and sulfur clusters. And that's it. And they also have a role in apoptosis in case of, for example, huge, for example, apoxia, which is when you don't have oxygen in the cell, or when you have DNA damage. So that's when the mitochondria triggers apoptosis. And another important thing you guys have to know about the mitochondria is that they cannot be generated from nothing. You can't have nothing or just a bunch of molecules and just want to produce a mitochondria out of nowhere. So this is what we mean when we say we cannot be de novo. De novo means in Latin is that you cannot make it from nothing. You have to create new mitochondria from old mitochondria. And this is where you have fusion and fission. So fusion is when you have two mitochondria fused together for the purpose of rescuing a mitochondria that has been damaged. And fission is for separating a damaged compartment of an existing mitochondria so that it can undergo mitophagy. Mitophagy is just eating up a part of mitochondria that has been damaged, and it's mentioned in the book. And other functions include the fact that it can bind to an endoplasmic reticulum or a tubule of an endoplasmic reticulum, and this is very important because this helps it do three things. What are those three things? Is that if the cell needs ATP, the endoplasmic reticulum, as we mentioned that the smooth endoplasmic reticulum is the one that controls calcium, right? So specifically in sarcomeres. So what happens is that they will pump calcium ions through the tubules into the mitochondrial matrix, which is this middle portion, which is what all the enzymes are. And this calcium will activate the enzymes that are needed to produce ATP. Do you guys understand? And then you have lipid exchange, which is just the fact that you need some lipids from the endoplasmic reticulum to carry out metabolic processes in the mitochondria. And then you have mitochondrial fission regulation, which is the fact that when you do mitochondrial fission, you have to wrap a tubule around the mitochondria in order to break it, in order to split it into two parts, and that's it. So this is just a representation of mitochondrial fusion, mitochondrial fission. So we said fusion happens when you want to rescue a mitochondria that has been damaged, and it's also for ATP synthesis. So I've seen a conflict in the book is that they say that fission is the one that's for ATP when you have high ATP demand, but here in the diagram it says energy demand is what triggers fusion. So both of them contribute to ATP production. It's just that they do so for different reasons. So this is for rescuing the mitochondria, and fission is when you want to produce more healthy mitochondria for more surface area, for more ATP, and to eat up and destroy the damaged mitochondria. So that's it for my part, and I hope I didn't take too much time, guys, and I hope you guys understood. So yeah, does anyone have any questions? Any questions at all? All right, thank you, guys. So I'm going to be handing the floor to my colleague, Mustafa.
Okay, Assalamu alaikum everyone. Bismillah ar-Rahman ar-Rahim. I'm Mustafa Al-Ani, and today I'll be giving you about the introduction, chapter 1 of your biochemistry book, your biochemistry syllabus. Inshallah, it's going to be easy. However, before I start, I want to make two things clear. First of all, the method that I'm going to be using, I'm going to be giving questions and you will be answering. Sadly, the sweets are almost all over. I think only 2 more sweets. So the first 2 questions, I guess, whoever answers will get them. And for the last part, for the actions, I'm actually going to explain stuff and then we're going to ask questions. The second thing, if you have anything in mind that you've studied and you didn't yet understand, please tell me now so I can integrate it into my presentation. So everything is clear, hopefully, in chapter 1. خلاص ليش جاي? Okay, so let's start. Let's check if you actually understand. So what are the functional groups that you can see in this molecule? Raise your hands. Now. That is correct. Yes, hydroxyl and aldehyde. But why is it? Yes. Clap, clap. أيوة. Yes. It's on the other side. Yes. So is it not a ketone? It's yeah. It has. Yes, it could be. So it has to be, if it's a ketone, it has to be connected to another carbon. That's correct. بس هذه يجيب الحلاوة يا إخوان. شكرا. أعطيه. Okay, next question. Just before we go to the next question, these are the functional groups. Now, I'm not going to go over all of them. I'm just going to go over the main points. So what makes functional groups polar? Who knows? Yes? That's true, yes. In general, if you have any electronegative group, which is something that one of my colleagues have explained, so one of the atoms would pull the electrons more than the other atom, and it would just take it for it. So in covalent bonds, you usually share electrons, but in this case, the share is unequal. And this creates polarity. So one of those molecules will be more negative than the other. One other thing I'd like to ensure is that aldehydes and ketones are considered isomers. And what I mean by that is that they have the exact same chemical formula. They are isomers in the sense that the molecules are arranged in a different way. And who can tell me what's the relationship between esters, amides, and carboxylic acids? Yes? No, not really. So what's the chemical relationship between them? Yes, you can say that. So. Yes, you can say that. So carboxylic acids give rise to amides and esters by different reactions that we have. Okay, so the next question. The three-dimensional structure of protein, what decides it? Yes. That is correct. Yes, can you explain, please?
So some like that are polar or non-polar, and acid and base. So it's depending on the hydrogen they form several.
Yes, that's absolutely correct. You are amazing, Salaha. Amazing answer. So in the book, they explained it in the way that the R groups are the determinants of the 3D structure. So this is just an reiteration of the answer. The sequence of amino acids is the sequence of how the R groups are arranged. Okay, so next question. Which macromolecule is involved in the storage of genetic information? Yes, that is correct. So what about the other options? What are their functions in the cell? نعم. بس خلي كمل أول. Yes. That's true. Yes, exactly. So as my friend said, carbohydrates and lipids are both for energy and structure. For energy, it's more mainly carbohydrates. However, for proteins, they have multiple functions. They have, I think in the book, you'll later take it, inshallah, in chapter 5, they have 9 functions. and you have to understand them. You don't really need to memorize them, but you, inshallah, once you get to it, it will be easy. So, the major carrier of chemical energy. So, the cell has energy. It stores energy in what form? Just can you raise your hand? Yes? That's true. It's ATP. So, all nucleotides with their triphosphates can store energy. But in the main one is ATP. So you can have GTP, CTP, and TTP. You can also have UTP, but they are always translated into ATP, so it's considered the main storage of energy. So for this question, when a region of DNA must be repaired, what causes the two strands to be connected to each other? I know it's a long question, so take your time with it. Yes, that's correct, but can you explain why it's not B? It's true, but generally the correct base pairing is done by hydrogen bonds. It's not a chemical bond, so it's a physical bond, so it doesn't require enzymes. It doesn't require that much energy to create. It just happens. It's like Velcro. Okay, so for the next question, when you form complex molecules from smaller ones, what is the type of metabolism involved in that? New hands. B, anabolic, that's correct. So what's catabolic? So anabolic it has another name. What is it? Someone said synthesis. Who said that? Yeah, it's called biosynthesis. So it's very close. Yes, you're absolutely correct. What about amphibolic? Does anyone know what amphibolic is? I don't expect you to know it, but it's nice information. You'll take it later, inshallah. It's both catabolic and anabolic. So it's a mix of the two. Okay, so for this question, it asks for the number of peptide bonds. Yes, that is correct. And how many proteins are there? How many residues are there? 3, that's correct. So it's always the number of bonds plus 1 to get the number of residues. So how did you know that it was 2? How did you count it? Yes, that's correct. So you mean those two, right? Okay. So they always are in a similar pattern. They always start with N and then O. So this is an easy way. And everything in between them is the amino acid. It's the R group. To know the number of residues, amino acid residues. So the number of amino acids in the protein. Okay, so for this question, it asks for the type of sugar. Yes. Aldohexose. So how did you know? That is correct. There are 6 carbons and there is an aldehyde, so an aldohexose. So what's an example of an aldohexose? Yes, what about an aldopentose? Yes. What about a ketohexose? Fructose. And I don't think they mentioned any ketopentose in the book, right? They did not. So one of the examples could be ribulose, but you don't need to know this. So this is a question that I noticed many doctors in biochemistry, they love to ask it in the introduction, but they never got the chance to put it in quizzes. So who would like to answer it? Yes? That is correct. So what about the other options? What are the polymers that they form? Yes, and carbohydrates. Yes, and nucleic acids. It itself is a polymer, actually. It's formed from nucleotides. So that's absolutely correct, yes. And for this question, I think it's the last question before we go into the reactions. The E. coli uses organic molecules both as a carbon source and an energy source. So what is the categorization of E. coli? Yes. E? It's not E. Waladi. Is it A? It's not A. It's B. You went all over the options until you used the correct one. It is B, yes. So what's the difference? Actually, let's go to the next slide. What's the difference between autotrophs and heterotrophs, first of all? Yes, that's absolutely correct. So your friend said is autotrophs, they take in, to be more specific, carbon dioxide. It's enough for them for growth, for carbon as a carbon source. However, for heterotrophs, they need to take in those macromolecules. So us as humans, we are heterotrophs. We take in carbohydrates, we take in lipids, we take in proteins. We cannot make them with our own. But what about photosynthesis and chemosynthesis? What's the difference between them? Another person. Minhaj, anyone? That's correct. Yes, the energy source. So phototrophs, they use light. Light is enough for them to break down those molecules to create glucose and then break it down, oxidize it into the energy form that they want. However, chemotrophs use chemosynthesis, so they need other organic molecules to break down those molecules and use their energy. So I hope everything was clear for the first part, because we're going to enter in the reactions. So I know I've heard before that reactions was kind of the difficult part in chapter 1. Is that correct? Okay, so inshallah after this session, everyone will know what reactions are and all the types of them and how to distinguish them. So starting off with nucleophilic substitution. So a nucleophile is generally negatively charged species, and it looks for a nucleus. Why a nucleus? Because a nucleus is positively charged. As you know, in an atom, there's a nucleus, and around it there are electrons. So the nucleus is made of protons and neutrons. So it's positively charged. If it's a nucleophile, it has to be negative. It has to be the other charge. So it's a negatively charged that looks for, therefore the name phile. I know a lot of my colleagues said the word phile. It wants positively charged species. And it's the opposite of an electrophile. So as you can see in the general example, in this case, A acts as the nucleophile, and it attacks B. Why does it attack B? Because X took its protons. So in this case, B is an electrophile. It's the positively charged group because the electrons of it are taken away, and A seeks the opportunity and comes in and attacks B. And this is nucleophilic substitution. One of the examples of it is hydrolysis, where in hydrolysis, the nucleophile is the oxygen of water. In water, the water molecule splits into OH minus and H plus. So the nucleophile is the OH minus, and more specifically, it's the oxygen of the OH minus, because it has the electrons and it wants a positively charged species to act on it and to become one with it. This is a deeper example of it. So it's the same. You just apply the general formula on this. Forget about this. It's not very important. But for this one, it's the nucleophile, and it attacks the carbocation, which is the positively charged species. And this is the part that goes out. So as you can see, this OCH3 was the X here in this case. It took its electrons and it went away, and it's left a space for the OH minus to come in and attack the electrophile. So the next three reaction types, they're quick. They didn't explain a lot of it in the book. Elimination is generally when a molecule is removed from a larger one, and the most famous example is dehydration. Usually what happens is it yields either a double bond or a triple bond, and sometimes in rare cases it can cause a cycle of a molecule. But most famously dehydration. As you can see, this is one of the reactions that happen in glycolysis. It involves the removal of water, so it's dehydration. For addition, it's the exact opposite of elimination. You break down a double bond or a triple bond, and you make it a single bond. And the most common example is generally hydration, and it always forms two smaller molecules from a one larger molecule. Yes. It could be reversible, yes, as you can see from. Yes, so they are the reverse of each other, basically. So you can see here that this goes this way, that's addition, and if you go that way, it's elimination. Yes. For isomerization, as I mentioned earlier, isomers are molecules that have the exact same chemical formula. The only difference between them is how the molecules are arranged. So in this case, you will always have one reactant, one product. You cannot have any more, any less. It has to be one by one. So in this case, as I mentioned, again, it's aldose and it's ketose. So you have an aldehyde and a ketone. They are isomers almost always because it's the same exact formula. So now for redox. Redox is probably the least explained. In many books, they usually write entire chapters about redox. Sadly, in the book, they only wrote a very small paragraph about it. Redox is generally reduction and oxidation. It's a mix. So it generally involves the transfer of electrons from one species to another. And because electrons are very small, you don't usually see them in the reactions. They're not seen while the reaction is happening or in the formula of the reaction. So instead, what students do and what scientists do is that they look for other signs, and they are the transfer of oxygen molecules or the transfer of hydrogen molecules into or out of the cell. So it's oxidation if you gain oxygen or lose hydrogen or electrons, and it's reduction if you lose oxygen or you gain hydrogen or electrons. And generally, the molecule that gets oxidized is called the reducing agent. Why? Because it gained an oxygen. That gets reduced is an oxidizing agent. So because it loses an oxygen, so it has to give the other molecule the oxygen. So it's called an oxidizing agent. So we say it oxidized it. And the same goes for a reducing agent. It's also the opposite. In the human body, usually NADH and FADH are used as reducing agents in the body to complete the reactions in it, and in multiple organisms, not only in humans. You'll take it later, inshallah, in chapters 8, 9, and 10 in the second semester. It's a very interesting story about it. You'll love those chapters. So those are the reactions, the redox reactions that could be given. As you can see, for this one, you cannot always see the reducing or the oxidizing agent. You can sometimes see only one of them. So this reaction is oxidation. You cannot see the oxidizing agent because they usually just omit it. Sometimes it's either very complicated or very unimportant, so they leave it out. This is just the opposite of the reaction, so it's reduction. And the last one is a reaction that happens in the body. It's the oxidation. It's the reduction, of lactate. And as you can see, it involves NADH, which, as we have mentioned, it's the reducing agent of the body and of organisms. So this is a quick review of the reactions that we took. Of course, you cannot take this as a 100% way to solve those questions correct. These are just starting points, so they can be wrong, especially for redox, because for this reaction, as I said, they don't always show it, so just as you've seen here. However, for isomerization, it's always 1 by 1. For addition, it's always 2 by 1. Not always, there are exceptions, but generally 2 by 1. Elimination 1 by 2, and substitution, you always have an electrophile and a nucleophile. Okay, yes. Yes.
How can we classify
Usually, as I mentioned, it's so redox. The definition is a transfer of electrons. However, we can't see it. So we see, as you can see, it gained an oxygen. First of all, it had one oxygen. Now it has two. So we can say it got oxidized. However, in this one, we had two oxygens and it became one only. Reduced, exactly. Okay, so now a question to test your understanding. What's the type of the reaction shown? Yes? It's not addition. Yes? No? It is E, yes. Why? Because it gained two oxygens. Now, why we cannot say it's addition? Because we don't have water. So generally, if you see OH, the ratio of oxygens to hydrogens is not 2 to 1. As you can see, H2O is 2 hydrogens and 1 oxygen. However, it gained 2 hydrogens and 2 oxygens. So this extra oxygen had to come from somewhere else, aka an oxidizing agent in this case, which is called dilute alkaline KMnO4. It's written up. Now, someone said isomerization. As I said, sometimes they write it above the arrow. Sometimes they don't even write it. So as I said, the number of reactants and products is not always an accurate way of telling it. It's just a starting point. It's fine. You can make mistakes. Now, as long as you don't make it in the exam. So what if instead of OH, you get one side that gets OH and another that only gets H? Exactly. So it's addition. What about if it was H and H? H and Yes, it is hydrogenation, which of the following? It is addition, yes. However, the many sources that I looked at, it classified it both as addition and redox because it gained H, so it got reduced. So for this reaction, can someone classify it for me? Yes? Exactly, hydrolysis. Can you explain it for me? Exactly. So you have two reactants and two products. In this case, the nucleophile is H3O plus, and it broke down this molecule and it took out the NH2 and it made it NH4 plus, and it substituted. It took its place. So now we can say it's hydrolysis and it's a nucleophilic substitution. Why is it not hydration? Yeah. Yes? To break down, yes. So you'd have two reactants, two products. In hydrogenation, you would break a double bond, usually, or a triple bond sometimes. Okay, so any other questions? Everyone understood everything? Okay, amazing. Yeah, that's it.
Thank you guys for